Good afternoon, everybody. Welcome to our 3-day online workshop on Genome-Wide Association Studies or GWAS. My name is Anusha Beers and I will be your moderator for the sessions. Before we get started, I take the privilege to introduce our speaker, Dr. Preenan Bagchi, an esteemed expert in the field of bioinformatics and currently serving as the founder, director of Basis Genomics Research Lab, Bengaluru. He has completed his postdoctoral research from University of Lorraine, France in Science and Engineering Research Pool. He has also worked as an assistant professor at Padma Shri Group of Institutions, Bengaluru and MGM University, Chhatrapati Sambhajinagar. Sir has earned his doctorate from Career Point University, Kota, Rajasthan, India in 2016. He holds certification in Ayurveda, Yoga and Sanskrit. During his doctoral research, he had attended IBRO-sponsored Advanced School of Neuroscience at Monash University, Malaysia. He has authored over 50 research and review papers in several national and international journals and in conference proceedings. He has presented his research works in conferences abroad. Sir has also guided several graduate and postgraduate students. He is an editorial board member of International Journal of Public Mental Health and Neurosciences, International Journal of Psychosocial and Cultural Genomics, Consciousness and Health Research, and Neuroscience Research Letters. Now I would like to introduce our second speaker, Mr. Harshit Reddy, an MSc Bioinformatics student at Reva University. He is a proficient in R, Python, and Linux environments and has an extensive experience handling cancer, microbiome, and next-generation sequencing data analysis. He is well versed in whole exome sequencing, whole genome sequencing, and metagenomics. He is skilled in automation tools such as Nextflow and SnakeMake and is currently conducting research on the microbiome. His interests lie in leveraging artificial intelligence and machine learning for bioinformatics applications. We welcome you, sir. Over to you, sir.
Yeah, Anusha, thank you so much for the introduction. Okay. Good evening, everybody. I hope my screen is visible to all and my voice is clear.
Yes, sir.
Okay. So we will start this workshop on this GWAS, that is Gene-Wide Association Studies. So there are many applications for this GWAS, especially in the area like diseases where you have mutation like diseases. You can detect exactly the where exactly is the disease occurring or the correct the loci and the correct SNP. So that is why this studies is very essential, and that is why this particular studies is needed. Okay. Okay, so to begin with, the GWAS, it aims to identify the regions, the specific region, or the specific region, we can say that the SNPs. So within the particular chromosome, we will have the SNP where exactly the disease is taking place or where the mutation has taken place. So if we can identify the exact region or the mutation, it becomes easy for treatment or if there can be any other therapeutics, we can go ahead. I just told from the human point of view. But if there is any, I just told from the human disease point of view. But if you have anything else, like if you want pinpoint particular SNP responsible for anything, any particular trait, maybe in plant or animal, we can do so many things. We can there will be so many other applications for the same. So that is why the study is very much essential. Okay. So aim to identify the which region or SNP in the genome are associated with the disease or certain phenotype. Mainly, as I told, the disease, if it is just the human disease mainly, and if there is any particular phenotype we need to identify, maybe in plant, maybe in animal, we can do it. So it helps to identify the population structure, select case subjects, that is those with the diseases, select control subject for the healthy, in align with the particular SNP, genotype a million SNP for each subject, and determine which SNP is associated. And finally, we need to go the entry and ranking for the SNPs. Yeah, means this particular study we are not concentrating on a single SNP, but actually a particular this which all the SNPs are responsible for the trait. So it is like, since it is a association study, there are how many or how many combinations of SNPs see, and we can understand it. Okay, so a short history for this GWAS. So successful study finished in 2005 with investigating patients' age related to molecular degeneration. You can see that in handbook of statistics, the first it was there, like editors being the Baldeling, Bishop, and Canning. So prior to GWAS in 2000, we had this inheritance studies of the linkage. Map was introduced way back in 2003. So these are all the short introduction, short history of this particular subject. And then the building haplotype maps for the gene findings. We have this human genome project. Then you have that is identifying the genetic variants and assay of the genetic variant. That is, we can verify the polymorphism, correlation, and anonymous with respect to the traits. So these are a few short history of this particular subject. So HapMap, you will be coming across this particular subject or this particular topic. So multi-country effort to identify the common human genetic variants. They need to understanding and catalog the LD, that is lineage disprium patterns across the genome in the several population. LD will be coming subsequent in the slide. The genotype, that is 4 million SNPs on the samples of like Africans, Asians, and others, it was done. And all genotypic data are available in a publicly available database, and able to examine the LD patterns, can estimate a given SNP. And sometimes it becomes difficult for us to get the SNP data, because in this particular it may not be always need to identify only the RS, but the group. Only RS we can directly get from the NCBI SNP or DB SNP, but for the like because we need to identify the bed file, BAM file, and so on. In the we will be telling you where all we may get the data, because sometimes it becomes little bit difficult or to get the data that is free data. Usually from the research papers we can get data for this particular work. Okay, so and there is a database. We will introduce the particular database accordingly. Okay. So why GWAS? So it is the tool to resolve the trait variants down to the sequence level. identifying the novel and superior alleles, then sequencing technology marked to the reduced cost. This is the genomic technology in which we do the sequencing, expression, profiling, and comparative genomics. And national diversity, that is harnessing the genetic diversity of the national population to the individual nucleotides. Okay, so why are they possible now? It is possible because we have now the genotyping technology, we are having it. Now we have the ability to type hundreds or thousands or millions of SNP in a single reaction on a SNP chip. The cost can be as low as approx $200 to 300 per person. We currently have the platforms like the Affirmatics and Illumina, wherein we can do this particular study. Initial days, it was like identifying the pinpoint, the loci, and it was difficult. Currently, we can do it thanks to the technologies which you have right now. So availability of the SNP, we have map and other resources to identify the SNPs and design the SNP chips, and further faster computers to carry out the millions of calculations to make the implementation possible. So how it is done, this will be a little bit of wet lab work, and finally you are getting the dry lab part. So as I told, the wet lab, the people, many researchers, they will be identifying or getting the SNP, and it will be published in the particular research paper. At instances, they can also put the particular data in the databases. So the cases like we have like case controls, and we are genotyping, we are getting the LD map, and from this we can get the Manhattan plot. So finally, like what we can do, we can get the SNP genotyping. Based on this, our analysis can be the LD, that is the linkage map, and we can identify where exactly we need to identify based on the Manhattan plot. We will do in the hands-on sessions, which will be happening tomorrow and the day after. Analysis of the GWAS. Most common approach, we need to look at each SNP one at a time, possibly add in multi-marker information, and further to investigate or report only the top SNPs. Finally, at the because we need to pinpoint which particular or particulars SNP, that is to identify top SNPs. Again, it is not single, it is multiple top SNPs, and or the backward replication, and commonly the trend test. Log adaptive models or the logistic regression we need to use and adjust for the potential population stratification. Basics of the GWAS to calculate the odd event ratio, the odd ratio. If two events are considered, the odds of A is like OR, that is A, or B, that is A, that is the A with respect to the approximation A, and B with respect to the approximation B. So that is the general ratio which we are following it. Significance, we can do it based on the chi-square test ranked by the p-value, that is the association test for the association. So we can search the SNP that deviate from the independent assumption and rank the SNP based on p-values. So for getting the exact p-values, we need to depend on chi-square and the statistics behind it. Here in this particular work, we will be using the R, that is the statistical tool, and we will be getting the SNPs which are we need for our work, for our work or for our understanding. So we are in the initial slide, we are talking about the LD, that is this linkage disequilibrium. So linkage disequilibrium, non-alleles, association of the alleles at the linked loci, a measure of the tendency of some alleles to be inherited together on the haplotypes descended from the ancestral chromosome. If these alleles, only the haplotypes in the population, then the alleles B and A, you can see that are in the pulphrase linkage disequilibrium. Here you can see the from the top line and then the below line, you can see that there is a disequilibrium. Equilibrium on the other, that is only the C and T are in the disequilibrium. It should have been G and A, but it has become C and T. So that is the perfect linkage disequilibrium. So that is to just to identify the pinpoint where exactly the difference has happened. So if we genotype the first SNP, we know what allele at the second SNP. Okay. The concept of the LD linkage disequilibrium, also referred to as the gametic phase disequilibrium or gametic disequilibrium, first described in 1917 by the Jennings, and this quantification, that is the coefficient of LD, was developed by the Newtonin in 1964. D is the difference between the observed gametic frequency and the expected gametic haplotype frequencies under linkage disequilibrium. So choosing the appropriate LD measures an objective of study. R square, usually we do it is the square of the correlation coefficient between the two loci. R square is affected by the mutation and recombination. Either it can be together or mutation and recombination, mutation or recombination. Either way, whatever may be the case. And other mutational histories. The R square varies between the 0 and 1. The R square value of equal to, that is 0.1 or 10% of the above are considered significant. So as I told here, in this particular case, we are using the chi-square, but since square will be implemented in the R, we will be directly get exactly the, or which exactly the SNPs we need. Okay. So factors in the LD, that is inbreeding, small population size, genetic isolation between the linkages, population subdivision, low recombination rate, population mixture, natural and artificial selection, and balancing selection. Other factors, outcrossing, high recombination, high mutation rate, etc. So there will be factors which can affect the linkage disequilibrium. So imputation of the SNP genotype. So we need to estimate the unmeasured or the missing genotype. So missing genotype. So we will come to know what is in the linkage with the phenotype. So if you are identifying which is the genotype which is affected, which are the phenotype which is affected, we can come and identify which is exactly the genotype which is affected and is expressed. So in this particular thing, we are actually pinpointing the genotype of the particular, say for example, the character. Based on the measured SNP and the external info, that is the haplotype population structure of the HapMap, we need to increase the power, actually mainly based on computation power of the laptops and the memory which we are using. So allow for the combining the data across the different platforms. So whichever platform data we are getting, that is Affymetrix or Illumina or etc., which are the platforms, we depend on that. Okay, here is one example like how we can what the observed genotypes can be with reference to the haplotypes. So you can see how it is aligned in the manner like you can see how it is aligned and what are the genotypes we are seeing and what we are considering. Okay, so here also you can see the missing genotypes which are all altered. So these are all, you can check it out here from the particular diagram. Okay, so advantages. So biological pathway of the trace have not been known. Biological pathways of the trace does not have been known. Discovering the novel candidate gene, that is very important. If it is say if it is a particular trait, or if it is a particular disease, if at all we are getting it, we are getting a novel gene candidate. That's a very excellent part. And because since I told a simple or single mutation lead to a novel gene, so single mutation, if you can pinpoint it, and if you are able to publish it based on the single where exactly the difference has happened, we are actually discovering the novel candidate gene. It encourages the collaborative consortia, rules out the specific genetic associations, where it provides the more robust data, identifies the mutations, explaining the few percentage of the phenotypic variant. So whenever you think that you need to, or you can do the GWAS studies, being a bioinformatician, you can find out the related GWAS research papers in wet lab or in the biotechnology, and you can establish a collaboration between the particular lab, the biotechnology lab, or whoever are doing the sequencing, and you can take the particular data and you can actually collaborate. That is, as I told, it is difficult to get the data online in the databases exactly, but if at all, if you are trying to get the data from the research papers, it is very good, and it gives a very good opportunity for collaborative research. Like if you are, it is easy to get the FASTA or FASTQ data of for the genomic studies, but when it comes to the SNP, this particular aspect is better. So limitations: the results need replication in independent samples in different population. A large study of population is required, detects association and not causation, identifies the specific location and not the complete gene. So this is a, though you can say that it is a limitation, but yes, but you are actually getting the specific location. So you need to focus on the common variant, and many associated variants are not casual. It can detect any variant in the less than 5% in a population. So cost of each DNA sample and pooling them, unavailability of the funding agency, that is always, it may be there, the constraints, and not productive and explain the less heritability. So based on the limitations, you can I understand the what opportunity is there in this particular area of study. In the genomic study, the GWAS plays a very important part, wherein you can actually do major collaborative studies. Okay, so will GWAS result explain more heritability? Possibly, if the causal SNP is not detected due to the power or the practical issues, that is, which is not yet included in the replication studies, and stronger effect for the causal SNP, that is, associated SNP may only serve as the marker for the multiple different causal SNP. Current association study challenge for the GWAS or the candidate gene. In the genome-wide screen, the hypothesis-free, high cost, that is large genotyping requirement and multiple testing issues. For the candidate gene, it is hypothesis-derived, low cost and small genotyping requirement, multiple testing and less important, that is possible, many misses, few false positives. And so why the GWAS is still popular, and it is actually very popular actually. So as I told that, first of all, the GWAS encourages collaboration in the between the labs. So that is why it is very popular. So the dropping genotyping cost, it involves the whole genome. So we need not to sequence the whole genome. That is the first point. So just exactly if you can pinpoint where exactly you need, or you need to find the trait, that is fine. So that is the 16S is also fine, or the just the SNP studies is fine. So resequencing of all the individuals in a population, large structure variation such as copy number variation, that is the resequencing of the arabidopsis. In future, this will help in the RNA-seq data to include the EQTL mapping in the GWAS study. Population choice will no longer restrict to the model organisms, will slowly become more focused on the SPP, which are more relevant in the answering biological questions. The accuracy depends on one-time genotyping and repeated phenotyping. Outputs. So all the genotype and phenotype data should be made publicly and be deposited in the public databases. Yes, people are doing it. Still, the data are, you can say, if you need exact data, sometimes it becomes difficult. But yes, as I told, there are databases are there. But if you need exact, sometimes you need to hunt and you need to get hold of the research papers. As such, file format and minimum information standard should be established, and priority to storage and dissemination of the phenotypic and genotypic data. One moment. I hope you are able to view my. Yeah, I think my screen is visible. Yeah. Okay. So in continuation, we are identifying the disease gene, that is identifying the genes that attribute to the disease risk is one of the main objective of the molecular research. Such findings have contributed to the improvements in the diagnosis, prognosis, and therapy with the successful identification of the disease genes for many single gene disorders. And many not single gene disorders, the rare disorders, it is the GWAS is becoming very, very important study. The focus has shifted to the disease with a complexity and multifactorial etiology, and the approaches, what approaches we can do after we are getting the particular linkage or particular SNP towards it. So, and how our genome can influence, it is given in this particular slide. You can check it out, that chromosomal abnormality and the mutation. So you can actually pinpoint where exactly the mutation has happened. So this is a very important study, because you don't need to actually do the whole genome and complete genomics. If it is a rare disease or thing, you can actually pinpoint it where exactly is that. So such kind of trained people are needed to identify or pinpoint the particular SNPs to identify the mutations mainly. Okay, so how our genome can influence the disease? So in various ways, like in several diseases like blood pressure, HDL, triglyceride, and so on, you can identify where exactly means our genomes are. You can know that they are influencing the diseases. But uh, if at all, if there is any particular mutation or if there is any particular loci, you can actually find it or identify it. That is it is a beyond, or it is deeper into the genome study. So identifying the disease gene, that is the association. So association refers to the co-occurrence of the genetic variant with the disease trait more frequently than can be readily explained by the chance. And two approaches to the association studies are like candidate gene approach and genome-wide approach. So this is the one particular it is mapped like the breast cancer patient and in the general population where breast cancer patient lie. So like if at all you are getting the whole genome, it is becoming difficult to identify where or what to search and where to. So the was important in this regard. So this is like you are having a much broader scope to identify the particular trait. So how is GWAS possible? To decide on the genotype, 12 million common SNP collect the 1000 cases with 1 to 1000 control. Genotype all the DNAs for the all SNPs that add up to 24 billion genotypes. Imagine this approach costs 50 cents of a genotype. That is 12 billion for each disease. So out of question. That is why can actually do this particular study pinpoint where exactly we need to go ahead. Okay. So this particular approach, we can actually get or discover the SNP from the particular chromosome. So you can see in this particular diagram the particular SNP which is pinpointed. So most common type of the genetic disability which is given here in the latest DPSNP, there are approx 62 million 76,000 and three SNPs are scattered throughout the genome, as you know, and abundance of the SNPs with the ease which they can be measured make these genetic variations significant. Common disease and common variants. You can always take this particular HapMap study. The common disorders are likely influenced by the genetic variation, also common in the population. Variants have small effects and multiple variants contribute to the disease susceptibility. Okay, heat map and linkage equilibrium. So in this particular picture, you can see to it that the SNPs are there, and if it is like it is based on combination or it is patients anyway, but anyway, our target is to get the SNP distributions. You can go ahead with the particular SNPs. That is one advantage of bioinformatics. As we are getting the our desired sequence, we can go ahead with identifying or pinpoint the particular SNPs. Okay. So this is again the HapMap project, wherein you are getting the European populations SNP, and approximately 1 million SNPs are scattered across the genome. So you have the technologies with us currently, Illumina, Affirmatics. We can actually tie up with the particular labs and we can get our sequence needed. So for designing the GWAS, so we need phenotypic measures, quantitative design, sample size and the power and population considerations. Okay, so in the cases we will have, you can see that cases in this particular, it will be less in comparison to control. So for our work becomes actually easy. So in the population, you can see the cases are less, while in the controls are more, and it is easy for us to with the study. So analyzing, so you can see we are getting the proper plots to we can identify the we can pinpoint the particular SNPs or the RS and how it is expressed. So analysis, the steps which will be following are the quality control, statistical quality. I told that you will need to do this chi-square test, the covariates and multiple testing. We will be seeing in the hands-on sessions which will be done tomorrow. So quality control, we need batch effect, that is SN, SNP quality control, that is missing HWE, MA, and all studies we will be doing it. And sample quality control, missingness, relatedness, and population structure. Statistical testing associations, that is generalized linear model approach, we will be doing it along with the logistic regression. So you can see that we will also be visualizing the Manhattan plots and the other regional plots. And we can identify the multi-loci analysis. Multiple testing, we will be doing it in the hands-on. And finally, we will be understanding the your work, and you will be getting pinpointing and getting the particular SNP which will be required for for the study. So finally, tomorrow we will be doing this hands-on for this particular GWAS. We will be using the SNP stats. Now I think Mr. Harshit will be taking. Anusha, I give the host back to you. You can ask Harshit to take over the session now.
Sure, sir.
Okay, any particular doubts you can you can put in the chat box. I will be in the meantime. When Harshit is taking the session, I can take over the questions in the subsequent time.
Hello.
Mr. Harshit, you can take over this session. Your host now.
Yeah, thank you. Am I audible?
Yes, yes.
Okay, fine. Great. Let's just start with the presentation. Hope my screen is visible.
Yes, it is visible.
Okay. Yeah, thank you. As discussed by Sir about the GWAS, now we are going
Mr. Harshit,
to,
can you put it onto slideshow mode?
yeah, 1 minute.
Yeah, thank you.
Yeah.
Yes, yes, great.
Okay. So as we discussed about the Jiva studies, now we'll understand about the tool that has been used for Jiva study. Okay. So starting with my background, I'm from bioinformatics background. I have MSc in bioinformatics. I'm equipped with R, Python, and Linux environments and other NGS analysis. In the automation end, I'm well versed with Nextflow and SnakeMake. My current research is about gut microbiome, and AI and ML are my interests. So yeah, we'll deep dive into the topic right away. Yeah, cool. So P-Link is a comprehensive GWAS toolkit. It helps in it's an open source tool set which was developed in 2007. So it is designed to help and favor genetic analysis focused on GWAS studies and population genetics. So it was developed by Sean Purcell in 2007. Let's look at the unique features of P-Link. So giving a forward about P-Link, P-Link exists as a standalone tool for command line options. We can use it from command line as well. But the best part about P-Link is it has R integration. That's R programming integration. So you can
Sorry
integrate
to interrupt, Harshit. Your slides are not moving. It is
slides and
on
not
the first
moving.
slide. Yeah. Yes, yes. Yeah.
Okay, fine. I think some technical glitch. Okay. So the best part of P-Link is it can act as a standalone tool in your command line. You can use it to shell or Ubuntu, Linux, or anywhere. Uh, Rather, more than that, you have access through R also. So P-Link has uh, specific R plugins through which you can access P-Link in your R or R Studio, R programming environment, right? So unique features of P-Link. Yeah, data handling. P-Link supports various processes down the line for GWAS analysis, that is starting from data merging, filtering, storage and processing, etc. And we can do quality analysis through this. We can check the quality of the data sets that you are getting. There are multiple parameters and options to do that, identifying minor allele frequency, genotyping rate, and other things. You can check the association testing as quoted by Sir in our GWAS understanding, QTL mapping, epistatus tests, and detection of SNPs are all favored from this tool set. Yeah, like other than GWAS, the more broader field will be population genetics. That is what P-Link is aligned for, like GWAS and the population genetics. Coming to how we are going to use this for our GWAS study, that's in the hands-on session. So P-Link R programming plugin support. So it has integration with R programming language. It can, as we all know, like R is known for its statistical analysis and visualization capabilities. You can perform all statistical analysis through R more efficiently than other languages. Like you have Python also, but then in bioinformatics, R is much preferred for statistics and visualization part. So how is this getting into, how is P-Link getting into R? It is through packages. There are specific packages called P-Link to R, GenAble, SNP GDS, PlinkSec, SNP Stats, etc. So through these packages, we are trying to we are trying to use P-Link toolset in our R environment, that is through R Studio, right? Then comes the data analysis. It helps in data analysis part in terms of bringing in statistical methods and data manipulation capabilities. Yeah, going forward, using P-Link with R for GWAS studies. We have various steps in doing the GWAS study. That is, first will be our data preparation. So we take specific genetic data sets and we perform quality analysis. Then we'll go for association testing, wherein we try to identify the genetic variants, followed by result interpretation. This is more based on statistics. You get the result data set and you try to visualize it. Advantages of using P-Link for GWAS analysis. So yeah, there are multiple brownie points for P-Link for GWAS. That is, like, it's very user friendly and it's open source. It's not and scalability. You can process too many data sets, large data sets within the P-Link environment, and it has community support. It has good community support. Since it's developed in 2007, the till 2015, there are updates in the versions, and there is a community platform wherein you can put your concerns regarding the analysis, and the community will help you out in terms of giving out solutions and stuff. Conclusion and key takeaways. So peeling, yeah, by the way. So this is the plot that you're seeing here is the Manhattan plot, which actually, as we discussed in our introduction to GWAS, the log 10 p-value is plotted against the chromosomal positions. You get Manhattan plot from this. We'll deep dive into this more in the hands-on session. So yeah, peeling is a go-to tool for GWAS analysis for various reasons. It is user-friendly, scalable, open source, and most of the research work that is going on, most of them for GWAS studies use peeling. Peeling with other softwares for like modifying their graphs into better ones. So yeah, in this slide you can see it's a circular Manhattan plot, which is a modification of your normal peeling Manhattan plot. So that way it is contributing to the research. Most of the research articles, uh, you can find these kind of plots. So for generating those plots, we need peeling and R. So we'll understand more about the tool and how it is supporting us in detail or more experimentally in our hands-on sessions, which is going to be on Wednesday, if I'm not wrong. So yeah, thank you.
Thank you, Arshad. That was a great presentation. Participants, I request if you have any questions, please post in the chat box as you're free to unmute yourself also. Sir, we have a question. I request you want to address the question that is in the chat.
So I think I am audible to all. So there was a question put in the chat box. It is, I just mentioned it. This is EQTL, that is expression qualitative trait loci. So what happens in this? When you are getting expressed RNA, so whenever there is the expression study you're doing it, you are getting the expressed RNA or the mRNA, which is transforming to the protein. So if you can detect the exact position of the expressed mRNA, then you can directly study the particular SNP with respect to the mRNA. So in that case, the particular study we are actually pinpointing again the particular our work, and we are taking it ahead. There are many research papers on this particular work also, and this is also very important part based on any genetic study. So I think I answered your question.
So there's another
Is it
question
I
from
answered question?
Niharika
Yeah.
Sharma.
Okay.
What are the exact input files needed for GWAS?
Yeah. There are for the GWAS, you will be needing bed. One moment, I will just start.
You need map files.
Yeah, bed beam the bed file you will be needing. Yeah, Harshit, you can go ahead.
We need map files as well. Optionally,
Yeah.
we need PED files. Yeah, most probably we'll use bed files and the map files.
Are the bed files used for a particular region, or what kind of bed files we exactly need?
It contains information like parental ID and all that. No, We are actually taking the raw bit files. We are not taking any processed bit files because processing comes in the later part, right? When we do quality control.
Okay, thanks. What kind of phenotypic data files are needed?
That depends on the data set or the study that we are taking. Let's say in certain population genetic studies or something like that, you need demographical data also. You need the color, height, all other factors also. So that depends more on the data set that we are taking for doing the analysis. In terms of GWAS, it will mostly be parental ID, family ID, and stuff like that. The history.
Do we need pedigree too?
Pedigree, no, like because in your bed file, it will have in your columns you will have all these parameters, right? Whatever the family ID and other things. In the rows, you will have individuals. So if you need pedigree and all, you can't process it. So all your files are basically in a format like Excel, right? Because to analyze statistically, it's more convenient like in that way. So if you take pedigree again, it'll be tough for you to analyze. You will add only those features, whatever features are there in the like in the pedigree, that will be added as phenotypes, different phenotypes in different columns, and in your rows you will have individuals, that is samples, right?
Yep. Okay, and how about, like, as I understand it, from the the SNPs that we need, so that means the genotyping files, the from the BAM file SNP calling should have been done. So is it the raw VCF file that comes as it is, or do you do some quality control on that SNP calling first and then use it for GWAS?
We need to do quality control. We definitely need to do quality control because if you are not doing like you can't rely on your study, right? At the end of the day, we need how it is associated. We need to identify the SNPs. We need to identify how it's associated or what is the location of that. For that, we need to do quality control. We will process it.
Okay, thank you.
Thank you, Niharika. Any other questions?
I had one for the EQTLs as well. So as Mr. Bakchi was explaining, so does that mean that SNPs called from the mRNA-seq dataset can be used in the GWAS studies as well?
Yeah, so your question was, does it mean that SNP from the mRNA-seq? So from the if you are getting the mRNA-seq data, but it has to be taken that if you are getting the specific data which is needed, that is the bed file or PAM file, if you are getting, then only it can be taken. But from the
From the
mRNA data, if at all you are taking the particular, I told if you are going ahead with the transcriptomic studies, from there you can go ahead in the particular transcriptomic studies and you can pinpoint the particular SNP. That will be again like two-way work. It can be possible, but you have to link it to further.
Yep. So my question was the SNPs suppose the SNPs have been called from the BAM files coming out from the transcriptomics study. So those SNPs can be used for the association studies too.
Which SNPs?
The like in the transcriptomics, the BAM files, if I use those BAM files and call SNPs on them, can I use these SNPs for association studies as well?
One moment. There is one moment.
Niharika,
So the
can
question, one minute. The
yeah.
question is like you're asking the integration of transcriptomic data or transcriptomic analysis. You will identify SNPs from transcriptomic analysis, that is gene expression levels. From that, you
Right.
want to integrate it to GWAS, right?
That's right.
Okay. So that can be done from your EQTL, right?
Yep.
So basically, that's what you're doing in EQTL. So
Yep.
when you get the data from your transcriptome, you get SNPs from transcriptomes that
Yes.
you will process for
Yep.
trait and loci like relation
Yeah.
to
So my
understand
question, yes. So my question is, can I use the SNPs called from the transcriptomic studies for the for GWAS as well or not?
Yeah, you can.
Mhm. And do you think that overlaying the SNPs called from the transcriptomics dataset on the genotyping dataset will give more power to GWAS?
No, I have to, like, we have to check that experimentally, like whether you are getting good kind of results or what kind of results you're getting when you integrate these both.
See, this is just a question. I have no experience in overlaying, but as per what my knowledge says, that if I suppose for the same trait, right? If I am investigating a trait, suppose in plants, and I've got the genotyping of a population done with a SNP chip, and I have transcriptomics data as well for that population. If I overlay the SNPs called from my BAM files coming out of the transcriptomics to the genotyping data set, will it give more power to GWAS?
That's what, like, experimentally we need to check that, right? What kind of results you're getting. We need to analyze the results. I personally have not done that. Or
Harshit, yeah. Harshit, I will just answer this. Harshit?
yes, sir.
Yeah.
Yes, sir. Go ahead. Go ahead.
Yeah, actually, since already you told it is the expressed one. So if it is, since it is the express, we are doing transcriptomic studies with the expressed gene. So definitely it will give the GWAS studies will get a accurate SNPs for this particular work. The answer is yes. It is a very wonderful study and it can be done, and we can get, actually it will boost the GWAS study. Yes.
Hmm. Okay. Thank you, Dr. Bakchi.
Hmm.
Any other further questions? Okay, if there are no, we can conclude the session. So I would like to thank Dr. Preenan sir for giving the comprehensive overview on GWAS study. And I also thank Mr. Harshit for explaining the peeling tool and its applications in GWAS. I thank everybody for your active participation. So we shall reconvene tomorrow at the same time. I request all the participants to join sharp 4:30 because the session is of 1 hour. So if you join as at sharply we can conclude it on time. So thank you, everybody. Have a great evening. Mr. Harshit, can you please end the meeting because you're the host?
Yeah, sure.
Yes.
Thank you, guys. See you tomorrow. Bye-bye.